Hi everyone. This is the fifth lecture for your chapter Photosynthesis in Higher Plants. In the last class, we discussed the light reactions. Specifically, in light reaction, we saw photophosphorylation, which is conversion of ADP into ATP, okay, synthesis of ATP in presence of sunlight, fine, with help of sunlight. So, in photophosphorylation, also, we saw two types. We saw cyclic and non cyclic. Cyclic produces ATP only and non-cyclic produces ATP as well as NADPH. We also saw chemoosmotic hypothesis which showed that how ATP is exactly synthesized. Okay, it is synthesized by producing a proton gradient. I hope you are reading along with me, and apart from reading you are also solving questions. Okay, the only way to gain more and more confidence is to solve more and more questions. Here you will gain that confidence, you will gain that much understanding also, so you will know where you lack and what areas you should focus on. Chalo, starting with the next part, which is biosynthetic phase. Before we start, you may have known that in 1945 the World War II ended. Okay, why did it end? It ended because America used the two nuclear bombs, okay, nuclear weapons against Japan in Hiroshima and Nagasaki. So after that, the World War ended. Now, during this war, most of the energy and research and resources, that is money and everything, was used to make weapons. Okay, it was used to make such nuclear bombs. But after World War, suddenly all of these radioactive materials and the knowledge about it was free to use. Okay, you can apply it to normal science as well. So, in this era, which was after World War, there was a scientist called Kelvin. Okay, this Kelvin was suggested by his other, what you can say, professor. Okay, this professor, he told Kelvin that you should use such kind of radioactive material in your study also, and that is how the whole reactions were found out. Before that, let us just briefly see what radioactivity is. Radioactivity is when the nucleus of any atom is unstable. It is unstable because the number of neutrons is higher than the number of protons. In such cases, the nucleus, it tries to become more and more stable. How will it become stable? So it can become stable by either alpha radiation, which is release of a helium nuclei. Okay, helium nuclei will have the two neutrons and the two protons. Clear? So that is called alpha radiation. This here is called alpha particle. Fine. So the nucleus can either be releasing alpha particles, which are nothing but helium nuclei. Helium nuclei I am saying because it will not have electrons. It will just have the protons and the neutrons. Fine. Okay, so that is alpha radiation. Then the second type of radiation is beta radiation. Beta radiation is nothing but electron which is getting made inside the nucleus. Okay, and it is released at high energy. So think about this. Neutron is neutral, right? It has zero charge. So sometimes that neutron gets converted into proton. They have the same mass and everything. So when neutron is converted into proton, which is positive, right? Let me just draw it here. This is the cursor I cannot. Okay, here, fine. So you have a neutron which has zero charge and it is getting converted into proton which has positive charge. So to balance this, okay, on the both side it should be balanced. So along with the proton, it is synthesizing beta particles. Beta particles have negative charge and they are nothing but they are electrons which are formed inside the nucleus. Because they cannot stay inside the nucleus, they are normally thrown out at high speed. So they are high energy electron particles or nothing but beta radiation. So the nucleus can also give out beta radiation to stabilize itself, or it can give out energy in form of gamma radiation. Okay, this is electromagnetic waves. in form of gamma radiation. Fine. What you have to know in our uh, whatever further process we have to study is this reaction. You have carbon, which has 14, carbon 14, as you may know, is radioactive, right? It decays. So it becomes into nitrogen 14, so N14, and gives out one beta particle. Clear? So as you can see here, in carbon 14, one neutron became one proton. So there is increase in the number of protons. From 6 it went to 7. And as you may know, atomic number is measured as the number of proton. Fine. And that is how you find out the element. So from carbon, the atom turns into nitrogen. Here the atomic mass remains the same. And this carbon, it gives out one beta particle. Okay. This beta particle again is nothing but electron. So this is the whole process. Now, as I told you, Kelvin was a scientist who was told by another professor or another scientist that they have a lot of such radioactive materials and these should be used for the studies. This is what he did. This is called a lollipop setup. Okay, as you can see, it looks like a lollipop, right? This is a handle, which you can see as a lollipop. Fine. So this here is the setup. You have this round flask, lollipop flask, which is filled with algae called chlorella. Okay, it is filled with green colored thing you are seeing is because it is water plus chlorella. Fine. It is filled with chlorella. Then here you can see a stopcock, and this here is the beaker. Fine. At the top it is again attached to multiple tubes, as you can see. Then there is light provided from both sides such that they will fall on this chlorella. Clear? Okay, let us see now what is happening. When the light will fall on the chlorella algae, it will do photosynthesis, right? But what this scientist did, what Kelvin did was, he entered radioactive carbon dioxide. Okay, from the upper tube, he will enter radioactive carbon dioxide. As we saw, for photosynthesis, you also require carbon dioxide. So these algae, they fixed that radioactive carbon dioxide and were producing radioactive sugars. Fine. How will you find out that they are synthesizing sugar from the carbon dioxide? So at some different time periods, okay, you have a timer also. At different time periods, you take some sample out of this lollipop flask. Okay, you take it in this another flask. You take the sample, you analyze the radioactivity and find out what kind of molecules have radioactivity after different times. Okay, so he checked. This is the graph after 5 seconds of light. Okay, after 5 seconds of photosynthesis, and this is the graph after 30 seconds of photosynthesis. Wherever you see a dark patch is showing more radioactivity. Okay, so because now that carbon dioxide would have entered, it would have gotten fixed in some other molecule, okay, other chemical, so it will transfer its radioactivity to this other molecules also. So which molecule is getting synthesized first? That was found out from these type of experiment. As you can see, after 5 seconds, there is somewhat PGA, some sugar phosphates, some sugar diphosphates, some other molecules also synthesized, okay, small molecules. After 30 seconds, you can see a lot of PGA is synthesized, right? Because it is showing you the darkest patch. So the more radioactivity is present where? It is present at PGA. So let us see how the whole process is happening, and this was the Kelvin experiment. So we move on to the biosynthetic phase, which is the next phase. Okay, I hope this much intro was enough. You will be able to understand this properly. So here is Kelvin. Apart from Kelvin also, there were two more scientists called Benson and Bassam. Okay, so this cycle that he gave is called Kelvin-Benson-Bassam cycle, though we just refer to it as Kelvin cycle. Kelvin, he used radioactive C14 in algal photosynthesis, and what was his study? He worked out the complete biosynthetic pathway. Okay, For example, let us say after 5 seconds PGA is higher. After 10 seconds some other molecule is showing higher radioactivity. After 5 seconds some other third molecule is showing higher radioactivity. So you know from which molecule is being made. Okay, for example, from PGA triose phosphate is being made, and so on. So this is how he found out the whole pathway, and hence it is called Calvin cycle. His work, his experiment, it led to the discovery that first carbon dioxide fixation product. Okay, now this is a new term. What is fixation? When you pick up carbon dioxide from atmosphere, okay, and convert it into some chemical, organic chemical molecule, okay, that is known as fixation. You are taking the carbon dioxide from atmosphere and you are fixing it in some other molecule or some other compound. This is called carbon fixation. Clear? So he found out that the first product of carbon fixation, that is the first time when carbon dioxide from atmosphere it is added to some other compound, some other organic compound, that first organic compound was nothing but 3 carbon organic acid. Okay, it had 3 carbon, it was an organic acid. The name here is 3 phosphoglyceric acid, PGA, here, which we saw here. See, this is PGA. After 5 minutes there is little bit of PGA. After 30 seconds, sorry, 5 seconds, okay, after 30 seconds there is a lot of PGA, and this PGA is getting converted into different other molecules as well. Fine. So Calvin was the one who found out that the first carbon fixation product, what happens when the plants take up carbon dioxide? The first product it forms, the first product plants form is a 3 carbon acid, which is 3 phosphoglyceric acid. Clear? He won Nobel Prize. Sorry about that. So, right, so these are the two stable products, okay, 3-phosphoglycerate and oxaloacetate. If these are the products, what is the substrate? Okay, one substrate we know is carbon dioxide, but the other substrate will be the carbon acceptor molecule, something which is accepting the carbon, right? So people used to think that because the first stable molecule, which is phosphoglyceric acid, is a 3 carbon compound, that the acceptor must be a 2 carbon compound. Okay, there is some 2 carbon compound which is accepting 1 carbon dioxide and producing this 3 carbon compound called phosphoglyceric acid. However, after years of research it was found that the first carbon acceptor, okay, the carbon dioxide acceptor, is not a 2 carbon compound. It is actually a 5 carbon ketose sugar called ribulose bisphosphate. Okay, remember in the biomolecules we had also seen ribose and ribulose. Ribulose is nothing but it is a ketopentose sugar. Okay, it has 5 carbons. It has a carbonyl group or ketone group on the second carbon, and on the first and the last carbon, that is first and fifth carbon, it has 2 phosphate groups. Fine, so that is why it is called ribulose bisphosphate. Why it is not called ribulose diphosphate? Let me just draw the structure very quick. So you have 1, 2, 3, 4, 5. On the first carbon you have a phosphate group, and on the last carbon, fifth carbon, you have a phosphate group. Because these phosphate groups, they are attached to different carbon number, okay, carbon number 1, carbon number 5, this is called bisphosphate. Okay, so if you have, let us say, this carbon having one phosphate this side and one phosphate on another side, then you call it diphosphate. Okay, when attached to the same carbon, you can call it diphosphate. When attached to different carbons of the same molecule, you call them bisphosphate. Clear? So that is why the name is ribulose, which is a ketopentose sugar, bisphosphate or ribulose 1,5-bisphosphate. Its short form is RUBP. Clear? So unexpectedly, scientists found out that it is not a 2 carbon, but it is a 5 carbon compound. So after this 5 carbon compound, it will accept one carbon dioxide and become 6 carbon compound. The 6 carbon compound is very unstable. Okay, you will rarely find it. It is very unstable, and it will directly break down into 3 carbon compounds. Okay, so 5 carbon ribulose bisphosphate accepts carbon dioxide, becomes a 6 carbon unstable product. This 6 carbon product will directly break down into 3 carbon products. Okay, 2 molecules of 3 carbon product, which is nothing but phosphoglyceric acid. Fine till here? Okay. So Calvin and his co-workers showed that the pathway operated in cyclic manner. They also showed that whatever RUBP was used, okay, we saw here the 5 carbon RUBP is getting used. After the whole cycle is getting over, this RUBP is regenerated. Okay, you get back the RUBP. So Calvin pathway, it occurs in all photosynthetic plants. It does not matter whether they have C3, C4 or any other pathway. This is an important point to remember. Calvin pathway or Calvin cycle is seen in all the different plants. Okay, there is no difference. It is seen in all different plants. Whether it is C3 plant, C4 plant or any other pathway it is following. Fine. The Calvin cycle itself, now let us discuss the whole cycle which Calvin and his co-workers found out. It occurs in 3 main stages. First stage is called carboxylation, which is addition of carbon. Second stage is called reduction, which is addition of hydrogen. Okay, first you have carbon dioxide which will get added, then that carbon dioxide should be reduced, right? And the third part is regeneration of RUBP. Clear? Carboxylation, reduction and regeneration. Let us start with the first step, which is carboxylation. Carbon dioxide is utilized for carboxylation. Carboxylation means addition of carbon dioxide. So carbon dioxide is used for carboxylation of RUBP. This occurs through an enzyme called RUBP carboxylase oxygenase, okay, Rubisco in short. Fine. So it is ribulose bisphosphate carboxylase oxygenase, Rubisco, which results in formation of 2 molecules of 3-PGA. Let us see the whole reaction. This red colored thing is your enzyme Rubisco. Okay. This 5 carbon, 1, 2, 3, 4 and 5. 5 carbon compound is your RUBP. This here is your CO2. Clear? So now let us see what the reaction is happening. The RUBP will come and attach on the active site. The carbon dioxide will also come and Rubisco will form a bond between this carbon dioxide and RUBP. But this product, okay, this 6 carbon product, 1, 2, 3 and 4, 5, 6, this whole 6 carbon product is not very stable. So as soon as it leaves Rubisco, it turns into 3 carbon products, okay, 1, 2, 3, 3 carbon together and the other 3 carbon together. These 3 carbons are nothing but they are 3-phosphoglyceric acid, okay, which is the first stable molecule formed after carbon fixation. Now this whole thing, this whole cycle, it happens 6 times. Okay, so 6 carbon dioxide molecules are required for production of 1 glucose. Glucose has 6 carbons, right? And if you remember, we said RUBP is getting regenerated, so you will require 6 such carbon from carbon dioxide to form 1 glucose molecule. Hence the cycle should happen 6 times. There will be 6 RUBP plus 6 carbon dioxide, which will give you 12 3-phosphoglyceric acids. Okay, 12 molecules of 3-phosphoglyceric acid. Second reaction or second phase is reduction. Reduction is a series of reactions that lead to formation of glucose. Now carbon dioxide has been added, you have 3-phosphoglyceric acid. The second part is making glucose. How will you make glucose? So just pay attention here on the diagram. You have 3-phosphoglyceric acid or 3-phosphoglycerate. This 3-phosphoglycerate will there will be addition of one more phosphate, okay, on the first here end, on the carboxylic acid end. So one ATP is used for one 3-phosphoglycerate. This ATP will donate its phosphate and become ADP. So now you can see there are two phosphate molecules, right, attached. So this forms 1,3-bisphosphoglycerate. Clear? From 3-phosphoglycerate, now you have 1,3-bisphosphoglycerate. This 1,3-bisphosphoglycerate will get reduced. Okay, what will be the reducing agent? It is nothing else but NADPH. NADPH will give its hydrogen, okay, and where will it give its hydrogen? It will give on the first position. So when it gives that hydrogen, the phosphate is removed. So here you can see Pi being removed. NADPH will get converted into NADP plus. The hydrogen will be transferred here, and now this molecule here is your glyceraldehyde 3-phosphate. Basic chemistry, if you want me to recall, whenever you have carboxylic acid or COOH on reduction gives you aldehyde. Aldehyde on reduction gives you alcohol, right? So here what the reaction is happening, this was COOH, right? So it was carboxylic acid group. This carboxylic acid group got converted into aldehyde group. So from glycerate or 3-phosphoglycerate, it became glyceraldehyde 3-phosphate. Clear? So this is how reduction is taking place. As you may have noticed, here ATP and NADPH are being utilized. This ATP and NADPH we got from the light reaction, okay, from cyclic and non-cyclic photophosphorylation. Fine. So 2 ATP and 2 NADPH are used for reduction of 1 carbon dioxide. Okay. So in this cycle here previously, if 1 carbon dioxide is joining, it is forming 2 phosphoglycerate 3-phosphoglyceric acid molecules, right? So for 1 phosphoglyceric acid molecule, you require 1 ATP. But because 1 molecule of carbon dioxide is giving you 3 phosphoglycerate molecules, 2 phosphoglycerate molecules, right? So you require 2 ATP and 2 NADPH per molecule of carbon dioxide fixed. Clear? I hope I'm making it clear. We saw that Rubisco is adding carbon dioxide to RUBP, and this process is giving you 2 molecules of phosphoglycerate, PGA. 2 molecules of phosphoglycerate will require 2 ATP and 2 NADPH to form 2 molecules of glyceraldehyde phosphate. Clear? Now, as I had already told you, there should be 6 cycles where 6 different molecules of carbon dioxide are added to make 1 glucose. So 6 carbon dioxide, 6 cycles are needed for 1 glucose, which means 6 into 2 will give you 12 NADPH and 12 ATP. Here, so in total to make 1 glucose, okay, for making 1 glucose you require 12 NADPH. Understand this, this is very important. Okay, remember this, for 1 glucose molecule you require 12 NADPH molecules. Fine. After reduction comes the third step, which is called regeneration. Regeneration again requires 1 ATP for 1 RUBP. As we saw, the cycle runs 6 times, so there will be 6 RUBP, so you require 6 ATP. Okay, and this ATP is used to regenerate the RUBP. Let us see this whole Calvin cycle a little bit in detail, okay, a little bit clearly, one by one. Okay, it is very simple, just understand it properly. Fine. We start with the carbon dioxide. So there are 6 carbon dioxide molecules required. I told you. Okay, 6 carbon will make 1 glucose. Fine. So 6 CO2, 6 carbons. This 6 CO2 will be added to ribulose bisphosphate. Ribulose bisphosphate is a 5 carbon compound. Okay, so it has total of 30 carbons because 6 molecules of ribulose bisphosphate. So 6 into 5, 30 carbons. 6 plus 30 carbons will give you 36 carbons. Clear? Fine. Let us just focus on the molecules now. 6 carbon dioxide plus 6 RuBP will give you 12 3-phosphoglyceric acid. Okay, 6 plus 6 is giving you 12. Okay, and this 12 is divided into 3 carbon compounds which are phosphoglyceric acid. So 12 molecules of phosphoglyceric acid. Now this phosphoglyceric acid we had seen in reduction that it gets converted into 1,3-bisphosphoglyceric acid. During this reaction 12 ATP are used. Okay, and you get 1,3-bisphosphoglyceric acid. This 1,3-bisphosphoglyceric acid in the second step of reduction, there will be addition of hydrogen from NADPH. There are 12 NADPH required. Okay, we saw that also for 1 molecule of glucose you require 12 NADPH. So this NADPH will donate its hydrogen and form phosphoglyceraldehyde PGAL. Clear? There will be 12 molecules of phosphoglyceraldehyde. From this 12 molecules of phosphoglyceraldehyde, you will have 2 molecules of this phosphoglyceraldehyde forming fructose, glucose, and other sugars. Okay, so 2 molecules from this 3-phosphoglyceric acid, 12 molecules of 3-phosphoglyceraldehyde. Sorry, you have 2 molecules which will get converted into glucose and fructose. Again, 2 molecules of PGAL means 2 into 3 carbon, right? These are 3 carbon compounds. So together 3 plus 3 will give you 6 carbons, and these 6 carbons will be forming the fructose or glucose. Fine. So from 12 molecules of 3-phosphoglyceraldehyde, 2 will form glucose, 2 will form sugar. The rest 10 will be used to make or to regenerate ribulose. Clear? So from this phosphoglyceraldehyde molecules, it will form 6 molecules of ribulose phosphate. Fine. From 10 molecules of PGAL. Again, how to remember? 10 molecules into PGAL is a 3 carbon compound. So 10 into 3 is 30 carbon. Ribulose we know is a 5 carbon compound, right? So 5, 6 is a 30, right? So it will form 6 molecules of ribulose phosphate. Now remember, we require ribulose bisphosphate. So for this, there should be addition of one more phosphate. For 1 molecule of RU, P, okay, ribulose phosphate, you require 1 ATP. For 6 molecules of RuBP, you require 6 ATP. And this 6 ATP will donate phosphate and make it ribulose bisphosphate from ribulose phosphate. And this is how the cycle will continue. Next time the carbon dioxide will enter, they will get attached to this ribulose bisphosphate and the cycle will again continue. Clear? Just pause this screen and draw this diagram in your book, okay? Because I know it will be simpler for you when you try to refer it. Okay, this here is the complete sort of diagram which is required for your understanding of Calvin cycle. Fine. From this cycle, let us now understand the balance sheet. What was in? What did you add? You added 6 carbon dioxide molecules. So 6 carbon dioxide in, 1 glucose molecule out. Fine, you are adding 6 carbon dioxide, you are getting 1 glucose out. You are adding 12 NADPH and you are getting 12 NADP plus out. These NADPH are used in the reduction reaction. Fine. Then you have ATP. So 12 ATP are used in reduction. Remember, for conversion of phosphoglycerate to 1,3-bisphosphoglycerate. and 6 ATP are used in regeneration of ribulose bisphosphate. Clear? So 12 plus 6 gives you 18. 18 ATP are used and you get 18 ADP. Clear? So this is the whole balance sheet of your Calvin cycle. You require 6 carbon dioxide, 12 NADPH and 18 ATP to make one molecule of glucose. Fine. So now finally you have glucose in your hand. Okay. And now the whole photosynthesis reaction will be very clear in your head, which is carbon dioxide plus water, okay, CO2 plus H2O giving you carbohydrate. Fine. So that whole reaction you will be able to justify now. Okay. Next time we will be talking about the different types of cycles. This what we saw can be included in C3 cycle, okay, because the first stable product was 3 carbon compound. Next we shall see that there are different kinds of plants also, as we have discussed C4. Then we will be seeing CAM, okay, cressolation acid metabolism and photorespiration. So see you next time.